Hello everyone. My name is Huate Li, and I'm a second year PhD student in Mechanical Engineering Department at BU. So, as a graduate research assistant in Thomas Stefano Lab, I am a personnel among Thrust Area 4, which is under Engineering Research Center for CellMat. So my research is mainly focused on photonics, which deals with optical imaging problems for biological applications. So here's an overview of my talk today. I will first give a brief introduction of some general terminology or technology in optical imaging, and then I will spend some time to talk about some current limitations of technology, and that's also the reason why we are spending so much time and effort in ERC, in CellMap to dealing with such issues. And after that, I will talk about share with you some current progress of us for optical techniques developing for CellMat, and then I will tell you our longer term expectations for CellMat. So here's the introduction part. So let's start with the simplest microscope, which you can see here. So a microscope makes an image of an object with an object here and a tube length here. And imagine that if you have an object like this, like an Air Force target here, and you really want to see what is the pattern inside this small area. And what you can do in this case, what you can do is that you put it under a microscope. We put it under a microscope, and you can see a expanded or enlarged image on the camera. So that in this way you can magnify the image that you want to observe. So, so no matter how complex or how complicated a microscope system is, that's the basic working principle of every microscope. And the magnification, which means how many times you can enlarge your image, the resolution, which means how clear your image is, and the field of view, which means the specific range you can see from your object in your image, are three main factors which characterize the microscope. And these three factors are actually controlled by specifications of these two optical elements. You can either change the magnification of the objective, or you can change the focal length of your tube length, or you can change the distance between them two. Okay, but things get a little bit different when it comes to biological microscopy. Since in biological microscopy, the object is three-dimensional. And in this case, we don't we want to see not just one plane of the object, we want to see deeper. And we want to expand our field of view to see the whole range of the object, which makes a challenge to the imaging. And another challenging part of this task is that when light passing through the tissues, the light will be distorted or aberrated, leading to a blurred image, which you can see here. So that's also something that we need to fix. So this problem has been a mainstream problem in imaging community, imaging or microscopy community, and many scientists, many researchers has developed many techniques to solve these problems, and especially for CellMat, our team has recently designed a technology called specially designed microscope called ASOM, adaptive scanning optical microscope, to fix this problem, which I will show it in the next slide. Show it to you in the next slide. So this is the ASOM. Basically, we get a wide field of view through increasing size of the objective, and using and which is combined with the stereo mirror. So with a certain tilting angle of the mirror, you can focus the light into different position of the onto the object. In this way you can scan over the whole area of the object. But one of the challenging part of this, one of the drawback of this technique is that since you are having a large objective, and inside this objective actually there are multiple larger lenses. So one lens will cause error to the system, and for multiple lenses the errors will be added up, and which result in an even larger error, so that the image you get may be blurred. So that's why place the deformable mirror here. Then through this device, we can fix the blurring with a computer command in a process called adaptive optics. And if you remember what I have talked about in the previous slide, um, you can see that you will know that we don't only want to see expanded field of view, we also want to go deeper into the tissue. So that's the second use of the deformable mirror in this setup, which I will show you here. So basically what we have done is that we push a pattern onto the deformable mirror to make it form like a curved shape, so that we can focus we can focus the light deeper into the tissue, allowing for an extended depth of field. And in this way, you can make a scan over the depth of the tissue rather than the area of the tissue. So this technique is called extended depth of field, or EDOF. So by combining these two techniques together, we have an extended depth of field adaptive scanning optical microscope, or ASOM EDOF. So during this setup, one of the important device we are using is a MEMS-based deformable mirror, or DM. The DM is the first initially developed at BU using silicon foundry processes. So basically, the structure of it is a reflective membrane, which is supported by an underlying array of actuators. So the way we control the surface shape of the deformable mirror is that we apply a voltage through these two plates, and by changing the voltage, we can the electrostatic force between these two plates are also changed. And since the electrostatic force is changed, the upper plate will either bend in or bend out, so that the surface shape of the mirror is also changed. So through this novel, fancy, small device, we can measure the microscope error, fix it, and also scan the focus with the DM. So speaking with CellMat, in CellMat, we are dealing with imaging problems with a more specific tissue called cardiovascular tissue, which are guided by multi-scale 3D engineered scaffolds. And for these tissues, they are first of all they are volumetric, which means they are wide and deep, and also they are highly scattering, which means the light passing through the tissue will be scattered to some random directions, and so that the the signal you get for the image will be degraded, or even you lose the signal. And the second thing is that since it is inside a heart tissue, it is inside a beating heart, it will be constantly moving due to contractile forces. And all of these factors will make imaging a big problem, a challenge in CellMat. So our challenges for ERC is that we want to get a high resolution imaging over large volumes, and also we want to get a real-time motion artifact-free imaging, which means we want to get rid of all the blurring image brought by the movement of the tissue. And then we want to do some external stimulation to the tissue to help it grow or differentiate. We can do it by either by optically, or by some external force like mechanically or magnetically. And we are in thrust area 4, which is optical engineering inside the ERC CellMat. So our main role in CellMat is that we want to build or design some novel, fancy, advanced optical tools for us to control and monitor cellular activities. And if we have these techniques ready, and the techniques in the other three thrust area are also ready, and we can incorporate them all together to build the CellMat test bed, and we test on it with some more complex nanosystem in living organisms, or even ultimately in human body. So that's my personal understanding of this three-plane diagram. And so to have a conclusion of our expectation, we want to build a microscope for volumetric imaging, which can of micro tissues to give a high frame rate, and we want to get a precise control of optogenetic actuation and fluorescence sensing in engineered 3D tissues, and we want to control the force applied to those tissues to help the heart tissue, to help the heart tissue or the cells to mature, differentiate, functionalize. So these can all be controlled by external forces. And overall speaking, we want to build imaging systems with high resolution to help us better understand what is really going on inside the cellular, in intercellular level or intracellular level of heart tissue. So finally, let me share with you some of our progress in CellMat in focusing optical side. So like I have mentioned before, we have just developed this ASUM EDOF microscope, which allow us to have a wide field of view of 9mm by 9mm, and also with an extended depth of field of 300 microns. Yeah, and actually we have tested on that with a small lens paper, and we get this really large, huge, giant image. And if you if you zoom in it a little bit into this small red area, the image you can got here is also quite clear, of high resolution, and you can see the inside structures are in different planes, which means it's so cool. Yeah. And another ongoing project is that we have a we have just developed a micro tissue incubator for force measurement and control. It basically we build a little box which is isolated from external environment, and we put a heater, CO2 and humidifier into it, and we construct a control system to control the temperature, the CO2 concentration, and the humidity inside the box to keep them all at a level where the micro tissue can grow or differentiate. And the second thing is that we just designed these tiny fixtures to fix which can climb down onto the PDMS wells, which contains the micro tissues, and through these fixtures we can either pull in or pull out those or stretch those PDMS substrate. And when you stretch these wells, the micro tissue, the microstructure inside the wells can also be stretched. So that's the way we give force to the micro tissues and do force measurement in the same time. And another thing here, another application ongoing project of us is that we just recently built a reverb illumination pulse ladder microscopy, and it is a new. I won't go too deep into this won't go too deep into some technical details into it, but I just want to say that it is a new technique to get many planes of images in the same time in a multi-photon microscope. And what you can see it here is that this left image is the image we got from a normal multi-photon microscope, and this image is the image we got from a reverb microscope. And you can see here that we can see more fluorescent beads through the reverb image, since we found out a way to let the beads, which is previously embedded in the background, to emerge. So in this way, you can see more beads and see multiple planes in the same time. The setup or the device I'm going to show you today is this microscope called ASUM, Adaptive Scanning Optical Microscope. So this microscope is quite elegant and designed device for optical imaging, in that so they have many optical components and has a complicated light path. So the thing is, so basically how it works is that you have a light source here, which is a blue LED, and there's a beam splitter here. The light will bounce onto the beam splitter and bounce to the mirror here, and travels to the objective here, and finally reaches the object that we are going to imaging here. So it is just a fluorescent bead slide, and after that, the light will be reflected back through from the slide here, and again travels through the objective to the mirror, and into this whole lens tube, and hitting onto the deformable mirror here, and then getting along this path, and finally reaches to the camera. So that's the whole working principle of the setup. And what makes it special is that we have this really large, extremely large objective combined with this steering mirror, and with a certain tilting angle of the steering mirror, we can look at different positions of the object that we are observing. So I'm going to show you that how something about how we can go to different locations on the object. So this software shows the image through that camera, and for now, the mirror, the tilting angle of the mirror is set at 0, so which means it stays at origin. But if I give it a certain command to let it tilt to a certain angle like this, it will be moved to another direction. And if you go to some other place, some other command like this will be moved to some other direction. And so let me first get it back to the origin. But as you can see here, even if we can scan over the whole area of the example here, actually the image quality here is quite bad. It's not that good. So that is because this objective is extremely large, and we have multiple lenses inside here, and the errors will and one lens will bring error to the system, and the error of multiple lenses will accumulate together and causes an even larger error to the system, so that we can get a bad image or a not very good image here. And the way we can solve this problem is that we added a DM here, which can fix these problems. So this is the image that we got from FlatDM, and you see that it is quite blurred. The image is quite blurred, and it's not the quality is not very good. But for this image here, the pattern are quite same since they are imaging the same location. But you can see quite clear that the beads, which is deep embedded into the object, you can see it now, and the initial beads, which is in the focal plane, can be much clearer now. So which means in this way we can optimize our image quality, while in the same time to see deeper. So that's our goal of this setup. Okay, so that's the end of my talk today, and thanks for watching, and I hope to deliver to you some more interesting or fancy stuffs in the future. Thank you.